Hello everyone, thank you for attending my presentation. I'm Hyunjun Choi, studying swine nutrition as a PhD student at NCSU. Today, we are going to study about the topic protein synthesis in skeletal muscle and jejunum is more responsive to feeding in 7 than in 26-day-old pigs. The paper was published at American Journal of Physiology 1996, and the authors were Teresa Davis, Douglas Brain, Marta Violotto, and Han Eugene. And their affiliation is United States Department of Agriculture. Let's talk about the introduction first. Protein synthesis in mammals is higher during the early postnatal period than any other period of life. And main part for protein synthesis in body appear to an elevated rate of skeletal muscle synthesis. This is because skeletal muscle had a large proportion of body mass and fractional rate of protein synthesis is high in skeletal muscle of early postnatal life. However, the mechanism mediating the development of change in the response of tissue protein synthesis to feed intake has not been established. Based on the previous finding, there are two main candidate factors to influence protein synthesis. First one is amino acid, and second one is insulin-like growth factor 1. In the previous study, amino acid increased the sensitivity of muscle protein synthesis to insulin inlet. However, the amino acid infusion alone or amino acids increase infusion of insulin to stimulate protein synthesis in human and pigs. So further investigation warranted to check the factor on protein synthesis. And IGF-1 is another factor. This is because IGF-1 is a regulator of effects of nutrient on tissue growth. Also, high IGF-1 in plasma and tissue decrease in response to fasting and dietary restriction. Based on this background, the objective of this study were to determine whether the response of skeletal muscle protein synthesis to feeding is elevated in neonatal pigs and to compare the developmental change in the response of protein synthesis to feeding in skeletal muscle with that in liver, jejunum, and pancreas. And lastly, to determine whether developmental change in response of tissue protein synthesis to feeding are associated with change in circulating content of insulin, amino acid, IGF-1. Here's the material method. For checking the protein synthesis in tissue, 7-day-old and 20-day-old pigs were used. And to compare the effect of feeding, those pigs were fasted for 24 hours, only feed for 3 hours after 24-hour fast. And jugular vein of those pigs were catheterized to collect blood sample for insulin, IGF, and amino acid content. And tissue protein synthesis were measured either the fasting or refeeding treatment period. Let's move on the results section. In this figure, x-axis represents treatment group age 7 day, 26 day, and feeding condition, fasting and refeeding condition, and y-axis represents Ks value. The Ks show the fractional rate of protein synthesis. To be specific, high Ks indicate high protein synthesis occurred. Let's interpret the result together. In this figure, the Ks value by feeding was greater in 7-old pigs compared with 26-day-old pigs. So the p-value of interaction showed significant between age and feeding on muscles and jejunum. So let's think about results together. Those results indicate that the degree of increase of Ks by feeding was greater in young age than older age in skeletal muscle and jejunum. The reason for this is likely due to increased insulin content. So let's see the result of insulin in this study. In this study, the feeding increased plasma insulin content in 7-day than in 26-day-old pigs. However, other factors, IGF-1 and amino acid, did not show interaction between age and feeding. The effect of feeding changed the IGF-1 and amino acid content. To be specific, fasting condition decreased IGF-1 content. Also, fasting condition decreased the amino acid content, and decline in amino acid with fasting condition was greater in 7-day than 20-day-old pigs. Based on this result, we can explain that the young age pigs can be more influenced by nutrients like glucose and amino acid. Let's move on protein synthesis capacity. In this figure, there were age effect on protein synthesis capacity in muscles, but not significant in jejunum, which indicate that the protein synthesis capacity in skeletal muscle is influenced by age effect. So let's think about results together. Those results indicate that the protein synthetic capacity in skeletal muscle is influenced by age effect. However, the protein synthetic capacity in jejunum is not influenced by age effect. The possible lesion is likely due to high elevation in protein synthesis in muscle than in visceral tissue during early life. This explains that muscle protein accretion show high in muscle than in whole body protein. For protein synthesis, we need to consider protein efficiency too. kRNA represents protein efficiency. High kRNA can increase protein synthesis. In this figure, the effect of feeding show on RNA in all part of the protein. So let's think about the result together. Those results indicate that the protein efficiency in most of protein is influenced by feeding effect. The possible reason is likely due to increase of protein synthesized per ribosome. Based on this result, we can conclude that stimulation of protein synthesis in skeletal muscle and jejunum by feeding is elevated in young compared with older pigs. Also, enhanced stimulation of protein synthesis by feeding in neonatal pigs is highly associated with elevation insulin content. Lastly, we can implicate those data energy source like glucose to improve insulin is required to improve the protein synthesis in muscle and visceral tissues that are related with growth of pigs. So let's summarize the paper content. The protein synthesis is in muscle and visceral tissue by feeding is elevated in young compared with older pigs. Insulin is important factor to improve protein synthesis in muscle and visceral tissue. And age effect on protein synthesis is more influenced by protein capacity. And feeding effects on protein synthesis is more influenced by protein efficiency. Thank you for watching my video. If you have any question or comment, just contact with me. And thank you so much.